Hello, everybody. Welcome to FAIR's free monthly webinar series. My name is Lynn Kewen. I'm FAIR's Senior Manager for Family Services, and I will be your moderator for today's presentation. I will be taking questions throughout the broadcast using the questions feature in the toolbar on the right-hand side of your screen, and if time permits, we will give our presenter an opportunity to respond to questions at the end of the presentation. As with all of FAIR's webinars, this presentation will be recorded and posted on the FAIR website in about 7 to 10 days. Please note that for maintaining a quality recording, all attendees will be muted throughout the webinar. Before we begin, I'd like to take a moment to introduce our presenter today, Dr. Jim Baker, CEO and Chief Medical Officer here at FAIR. Dr. Baker has more than 30 years of experience in basic biologic research, concentrating in immunology and host defense, both at the University of Michigan and in industry at Merck. He completed his undergraduate education at Williams College in Williamstown, Massachusetts, and his medical education at Loyola Stritch School of Medicine in Maywood, Illinois. After an internship and internal medicine residency at the Walter Reed Army Medical Center in Washington, D.C., he completed an allergy and clinical immunology fellowship, also at Walter Reed and at the National Institute of Allergy and Infectious Diseases in Bethesda, Maryland. His long-standing research in immunology involves the basis of several autoimmune diseases, and he has become an internationally recognized expert in nanotechnology, nanomolecular materials, and biomedical applications, and the emerging field of nanomedicine. His work with synthetic lipid and polymeric nanostructures has resulted in the development of a new class of antimicrobial agents. He also has developed a needle-free nasal spray adjuvant vaccine technology for influenza, hepatitis B, pneumococcal, and RSV. This work led to a startup company, NanoBioCorp, in Ann Arbor, Michigan. He also spent 2 years as the Senior Vice President and Global Head of Vaccines at Merck. For nearly 20 years, Dr. Baker was the Chief of the Division of Allergy and Clinical Immunology at the University of Michigan, where he helped start the Food Allergy Research Center. He also has served as the Ruth Dowdone Professor since 2003, and he retains his professorship and serves in an emeritus capacity at the university. Dr. Baker has authored more than 300 peer-reviewed scientific articles. So I'm very excited to have him with us to present to you today, and at this time, I am going to turn the webinar over to him.
So it's my pleasure to give this update about where we're at with food allergy and the science that underlies it. Food allergy for the medical community, unfortunately, has been an unrecognized epidemic. Despite the incredible increased numbers of young people with food allergies, many doctors really didn't understand the significance. And I think we've had a remarkable year at FAIR, not just in helping to define the scientific and medical basis of food allergy, but in also increasing the awareness in the medical community of this important problem. I think when we think about what is going on with the immune system in food allergy, we need to understand several things. The first is that no individual is born with their immune system intact. It's the only organ in the body that actually develops and evolves after you're born and can be shaped by experience. We've used this over the years to protect ourselves because by using vaccines, we could train the immune system to protect ourselves from certain types of infections. But because of this, everyone's immune system is unique, and it evolves in relationship to the environment. And on slide 4, it talks about some of the many inputs that can shape the immune system of young people. Just the idea that they have older siblings that might bring home infections or that they're in daycare really can alter their exposure to infections. The farming environment is now well known to expose young people to endotoxin and other types of things that alter their immune responses. Back when we were developing as a species, we lived in the primordial swamp where infections with parasites were very common. And remarkably, these types of exposures have changed over the years. And in fact, the idea that you have 6 or 8 children in a family, the idea that you have all kinds of infections that are shared, is very different from what the reality is now, where smaller families and urban lifestyles and more sterile environments occur. So in fact, one point I'd like to make to begin with is that human beings have fundamentally changed in the past few years, and that may underlie the epidemic of food allergy. The next slide, 5, is really the fundamental forces that have changed human immunity over the past 62 years. And I use 62 as a convenient marker since I was born in 1953, and you could do the math. But quite honestly, when I was born, my parents' biggest concern for me was that I might get polio. And any infection I got as a child was naturally acquired. There really were no vaccines to protect me from them. And the only antibiotic available was penicillin, and that had very little effect on the bacteria that lived in my gut. So in fact, it was a very different world back then than it is now. And medical advancements, environmental factors, life and lifestyle and diet changes, and genetic factors that evolve sort of epigenetically, which I'll explain in a minute, have all altered the human immune system. What's most remarkable is that there appear to be things that are inherited outside of genetics. And this is both in the environment of the fetus as it develops in the mother, and even before that, in the environment of the sperm and the egg that go to make the fetus. And those factors seem to be pressing us more and more towards an allergic lifestyle or expression in the individual. So when we look at the human immune system, there's been an incredible and very quick evolution over the past 50 or so years, 60 years, probably more so than in the preceding 500 or 1,000 years. And we think those factors are weighing in on whether or not people become food allergic.
I think we're going backwards.
Yes. If you look at slide 6, though, it tells you how complex this really is. This is from a review by Gideon Lack and Katie Allen from this year. And the red are things that may be risk factors for causing food allergy. The blue are things that haven't been associated, and the green are things that may be protective. So if you look at this, there are all kinds of different things. Just the family history of allergy in and of itself can cause this. There are genetic factors that are involved. There are different food elements inside the developing fetus and potentially even inside the mother as her eggs develop that may be very important. The country of birth may be very important, and in fact, people near the equator have lower evidence of food allergy than those farther away. Later introduction of allergic foods now appear to be important, and we'll talk a little bit about that and some of the things that we might have done that actually help predispose people to food allergy. And then things like vitamin D deficiency and exposure to food allergens on the skin may all be important in causing this. So this is a very complex interaction between environment and genetics that leads to a predisposition to be allergic. So I'm going to go through a number of questions that sort of frame the debate right now about food allergy, and try and as best we can, give you an idea of what the thinking is about this right now. And the first of these questions on the next slide is, can food allergies be prevented? And quite honestly, till this year, there was quite a bit of debate about this. And in fact, if you talk to pediatricians, and on slide 8, we'll go through this. Once they started seeing an increase in food allergy, one of the decisions that was made was to stop feeding young children food. And in fact, people pressed much harder on doing things like breastfeeding for longer periods of time, or they remove specific foods in the diet that may be prone to cause infections. And this started about 20 years ago. And what was really amazing was it didn't prevent the development of food allergy, even if people weren't eating it. And it became clear that one of the problems was that they were being exposed on their skin, especially if they had eczema, to food allergens, and that was sensitizing them. So even avoiding the food did not prevent food allergy. And in fact, Gideon Lack first noted at a conference when he talked about peanut allergy and went to Israel, he was told that there was no peanut allergy in Israel. And in fact, when he looked at the rates, they were much, much lower than that in England. And part of the reason was that peanut appeared to be fed as a hard candy for teething very early in life. So Gideon proposed to us about 8 years ago to do a study where he would compare children who were fed peanut very early in life with those that avoided peanut and see what happened to the rates of allergy. On the next slide, you see the end result of this. And this is a remarkable study. Gideon deserves, and his co-authors deserve, a tremendous amount of credit for this. But what you can see here is that in the groups that avoided peanut, which are in the gray bars, they had rates of peanut allergy that ranged from 13 to 35%, dependent on whether or not they were skin test negative or positive for peanut. So in fact, they had young people who already had skin test sensitivity, which suggested they had been sensitized to peanut, but weren't yet clinically allergic, i.e. if you were having allergic reactions to peanut or they challenged you with peanut and you had an allergic reaction, you were eliminated from this study. But in the young people who were given peanut and had avoided it, were given peanut within the first year of life, you could see that there was a remarkable reduction in the rates of peanut allergy. In the skin test negative groups, it went from 13% to 1.9%, about an 80% reduction. In the skin test positive groups, it went from somewhere in the 30s down to less than 10%. And so this really suggested that feeding the food early in life actually was protective against the development of food allergy, even in children who had already developed sensitization. So this was a very important thing to define, and in fact, it's already led to consensus reports from the American Academy of Pediatrics and a soon-to-be-released report from NIH suggesting that withholding food from young children as a way to try and prevent food allergy is not a good idea and may actually be detrimental. So if we now know that we could prevent this, this in the long run may be a very important thing as we look to different foods to see whether or not they too can prevent food allergy if eaten early enough in life. Right now there's no data about this except for peanut. So in fact, Gideon Lack is doing a study called ProNuts. It's 5 different foods, 5 different types of nuts, including sesame, looking at whether or not early feeding can prevent allergy there as well. But that data isn't out yet. And I think it'll be very interesting in knowing whether or not this can be generally applied to all foods. The other thing that's very interesting is, are these children just being desensitized, i.e. by feeding them the food, it's like oral immunotherapy where you just stop the food reaction, but you don't remove the allergy. And there's another study that should be coming out in the next year that FAIR also funded that will be looking at that effect and seeing whether or not early in life we actually tolerize or turn the immune system off to the food or simply desensitize by giving them the food. And that'll be very interesting as well. So, as I mentioned, The young people who are already food allergic were eliminated from the LEAP study, and many young people are allergic to food. So the real issue is, what are the treatments for food allergy? And I could be very snide and put up a slide that's blank, because right now we really don't have therapies for food allergy. In fact, the only thing we have that can treat people with food allergy is giving them epinephrine after they've had a reaction. And slide 11 talks a little bit about that. Epinephrine is the first-line treatment for allergic reactions or anaphylaxis. And that's a rapid allergic response that can lead to shock and death. Unfortunately, a couple of things have happened about that this year. We have two types of auto-injectors that could be used during these reactions. And unfortunately, one is currently off the market, the AviQ, and that's because they had some manufacturing issues with it. So we're left simply with the epinephrine injector, which is the EpiPen. The EpiPen is obviously expensive, and FAIR is going to try and work with Mylan, its manufacturer, and with charitable organizations like ourselves to make it more available to young people, because this is something that really needs to be given. But we also are working with doctors in emergency rooms, because some of them are under the misconception that giving people antihistamines or steroids are a treatment for acute allergic reactions, and that's not the case. Epinephrine is the only therapy that's available. So what do we have beyond epinephrine? What we really have as a goal here at FAIR is to develop therapies that can prevent allergic reactions to food in allergic people, i.e. treat them so that they don't have to worry about eating a peanut or coming across egg or other types of nuts in foods. And even if they do, they'll be tolerized enough, or at least controlled enough, that they won't have a life-threatening reaction. So we at FAIR believe that this is a pivotal time for drug development for food allergy. And why is that? So one of the reasons is that we have developed a couple of ways to advance these therapies much more aggressively than we have in the past. And in fact, some of the ways we've done this is to try and change the environment to develop new drugs for food allergy. And FAIR had two big advances in that this year. The first advance on slide 14 was the launch of our clinical network. Quite honestly, there weren't enough sites in the United States to even do clinical trials. And so FAIR undertook the effort to set up sites across the United States that would have a research coordinator and a trained allergist that could do clinical trials, that could do food challenges, that could do all of the difficult things necessary to care for allergic patients and to develop new drugs. This year, we for the first time had 24 sites across the country where these centers were set up. And they went through a rigorous vetting campaign to make sure that they had the capability to do this and that they were trained to do research. And these 24 member centers of excellence are really going to be important in the approval of new drugs, because for the first time, we will be able to do clinical trials with large numbers of patients. The next slide talks a little bit about what these clinical network sites can do. First, they're going to raise the quality of care for food allergic patients nationwide. They will reduce discrepancies in the quality of care between providers, because people are going to collaborate and share their best practices. They'll make comprehensive care available and accessible for all food allergic patients, no matter where they're located. Eventually, we go up to 40 or so sites to make sure that there are sites in almost every state and access to almost every individual that needs it. And finally, we look to bring new sites for clinical trials of new therapeutics so that it'll be easy for companies that want to test drugs for food allergy in a controlled way. Without this, everybody was doing things a little bit differently. And the nice thing about these fair clinical centers, they've all agreed to do things like the food challenges the same way and do clinical trials under the same rules so the data can be collated and will be useful to NIH. And they all also plan on sharing the data more readily so we can do large-scale trials and really know what's effective very rapidly. We also hope to build a national food allergy patient registry to make patients available to new drug trials. So individuals who want to participate and try new drugs will have first access to this nationwide. And you can imagine what a real hurdle it was to try and set up an approval trial for a drug if all the sites have to be collated, if they all have to be trained how to do each trial differently, if they all don't really even come from the same page. So this has been a real advance, and there are actually clinical trials that are being done right now under the auspices of the clinical network. The next slide gives you an idea of where the current sites are. You could see that they cross the nation, and they're in a number of states, and we really hope to have sites in every of the 50 states eventually, so that there'll be a place for everyone to go to. You could see that many of these sites are places where food allergy was not being done before, or that food allergy was not a priority for. All these centers have committed to supporting this, and we had to get sign-off by their chief executive officers for the health systems to document that they're really going to do the right thing for food allergic patients. So we're very proud of this. You can look on the side and decide where the closest center is for you, but we look at these places as a beacon that people can go to get help with food allergy. So the other thing we needed to do was actually just increase the number of scientists and physicians who are involved in food allergy research and care. And that's a big thing. Even though there are allergists that are trained to do this, there are very few in the country considering the number of food allergic patients are. And in fact, many of them were not doing things in a standardized way. So we were able to do that, and we're now bringing in new people. And we're bringing in new researchers. And we have 5 specific awards. They're mid-career awards, along with our Get Us Award for people in training, to increase the number of young and mid-career investigators in food allergy. And this is also increasing the diversity of investigators on our review and advisory boards. So the more different perspectives you're getting on food allergy, the better it will be in terms of trying to figure out what's the best way to proceed with new therapeutic approaches. And our next slide, 18, has the new investigators on it. Jessica O'Connick at the University of Michigan and Dwayne Weissman at Harvard. And our 3 mid-career investigators, Simon Hogan at Cincinnati Children's Hospital, Michiko Ohashi at Boston Children's Hospital, and Eric Wambra at the Benaroya Research Institute. We're very proud of these people. They competed with over 20 other national scientists who were the other finalists. So you can see it's a select group, and the quality of their research is very impressive. And I feel that several of these folks will have the ability to change the discussion about what's important in food allergy. Our Research Advisory Board has also been expanded. You can see there are a number of younger investigators here, Pam Guerrero at NIH, Nick Lucas at University of Michigan, Kathy Nagler at University of Chicago, and David Artis at Weill Cornell, who are really fundamental folks in their fields. David Artis does immune inflammation. Pam Guerrero does allergic inflammation. Dr. Lucas has actually been an asthma specialist who's now looking at the same type of allergic pathways in food. And Kathy Nagler is looking at the biome. So along with some of our holdovers, such as Bob Schlemmer, Stacy Jones, and Dan Edelman, and myself, this is really a different group of people. They're independent. No one who reviews the grants is involved in making any decisions with anyone they know or have worked with. So in fact, I think we've made our process much more rigorous, and the quality of the investigators we have demonstrate that. So what else can we do to accelerate new drugs for food allergy? I think the most difficult thing we have in food allergy is the fact that we need to do food challenges as our outcome measurement. There are 50 drugs in development for asthma, and the reason is they can do a breathing test and know whether or not they work. With the idea you have to feed a food to someone and see if they have an allergic reaction to know if it's working, you can imagine that's a much bigger problem to try and deal with. So one of the things we're trying to fund and trying to support is the ability to do a surrogate marker like a breathing test for food allergy in a way to try and define an outcome that won't require people to do food challenges. And we've now looked at a number of things, and it turns out that there's nothing right now that's really something that's a replacement for food challenges. And part of the reason we need the network is that we need a lot of people that are experienced and can do food challenges safely. It's the gold standard. If we could think of other things, that would be great, but in fact, it's a huge problem. And in fact, a study done by Immune as part of their control arm showed that there are psychological factors in food challenges as well, so that you have a bit of a placebo effect, and that also clogs the results. So in fact, you're going to have to continue to do food challenges. There are some hopeful things in research, but quite honestly, there's nothing right now that really shows a replacement for food challenges. So given all these challenges, the good news is there are finally things in the later stage of development to treat food allergy. And these are really remarkable approaches. They're things that FAIR has supported and continues to support, and we will support not only while they're in development, but we'll support their approval at the FDA, and we'll support getting third-party payers to pay for them, because we feel that once food allergy therapies are available, everyone that has a life-threatening reaction should be on them, because it's a very, very, very important thing. So there are two things right now that are in late-stage therapy that are antigen-specific or food-specific, if I could put it that way. The first one on slide 23 is the Viaskin peanut patch. And what the Viaskin peanut patch is it's a translucent, breathable membrane that has peanut allergy attached to it. And it's then put onto the skin with tape, and it's occlusive because of the breathable patch, and that allows the protein to actually penetrate into the skin, where it's taken up by dendritic cells or antigen-presenting cells and turns off the immune response to the food. And you can see here on the arm that this sticks on the arm. In fact, in most cases now, they're putting these on the back. But essentially what this does is if you wear it, it prevents the food, it prevents food allergies or builds up your tolerance to the amount of food you can eat. On slide 24, you can see that this is placed on the skin. As I alluded to, it's delivered to the immune system through dendritic cells. There was a 100-subject clinical trial that was done, and it was very interesting. The adults did not seem to respond, and anyone over 11 was eliminated. And the good news is that the young children between 4 and 11 years of age did respond and did develop increased resistance to allergic reactions from foods. So it was really very impressive because there were very few side effects other than a little eczema at the side of the patch. So this suggests that especially for young children, age 11 and below, the patch may be a useful thing. This is now in a large-scale phase 3 trial. They're talking to 300 to 600 patients. So you can imagine that's 5 to 6 times their other trial, and they need lots of patients. So in fact, you'll be hearing that this trial is going on many places around the world. I think they have 50 sites, including our clinical network sites. And please, if you hear this is available, if you have someone this age who's peanut allergic, please be willing to take part in the trial. The other therapy that's coming forward is from Aimmune, and this is oral immunotherapy. Now, OIT has been done for a long time by many allergists in the community, and it was very difficult to do. Most allergists didn't do it because there was no set way to do it, and there was no set amount of food that you needed. And in fact, there were concerns that if you used different types of peanuts or different preparations of peanuts, you'd be dosing people differently, and that would be a problem. So in fact, what Aimmune does, and Aimmune, for disclosure's sake, was started by FAIR and is now a public company, but they feed small and progressively larger doses of food to an individual with the intent of desensitizing them. And the dose of food is very consistent. What they've basically done is made food into a drug. So they've taken peanut, they've standardized it, so whether you get peanut from your local allergist or an allergist across the country, it's the same type and antigen load as the other site. And by giving this food, the patients who were able to tolerate the desensitization really had a significant reduction in their sensitivity to food. And it was fairly impressive. Now, the negative is that about a third of patients did not tolerate either the challenges or the increasing dosing of the food. So in fact, the efficacy was very impressive, but the side effects were also impressive. So this won't be for everyone. It may be for about 2/3 of the patients, and there may be other ways to get other people on board. But this is really, does appear to have significant efficacy besides the side effects. I can tell you that, this really, I mean, FAIR has actually supported OIT for quite a while. And I think what we've been able to do with this is get it done in a standardized way. So eventually, not just allergists, but potentially even other types of doctors could give it to patients. On the next slide, you could see the updosing, and you could see the patients are given over a number of weeks, an increasing dose between 1/2 of a milligram to over 300 milligrams that they eat every day. And by doing this, you turn off the allergic reaction so that if peanut is accidentally ingested, you don't have the life-threatening reaction. And you can see here this takes 24 weeks, so it's not for the faint of heart, and you have to go into the doctor every week to get this. Increased dose, then you can go home and maintain it at home. But it's a fair amount of effort. On the other hand, if you're one of the 2/3 of the people that respond to it, it's a big advantage. And slide 27 gives you some of the data from their clinical trial that's on their website. And you could see that the amount of peanut that's tolerated in the control group is significant compared to placebo. You know, it's actually 5 times what the placebo is. And in the entire analysis, you know, you're looking at people tolerating 440 milligrams of peanut protein. So it's a very impressive accomplishment when at the beginning they didn't even tolerate 3 milligrams. So, you know, this is a real advance, but it's an advance only for some of the patients, and we need to keep that in mind. And it only works for one food right now, so you'd have to desensitize to every food, and that's certainly a negative for patients who have multiple food allergies. So what else is out there? Well, it turns out that we have a much better idea of the immune system and what causes allergy. And on slide 29, this is a photo from Nature Reviews Immunology that shows the interaction of what we call the immune synapse. And over on the left, the purple cell is the dendritic cell, or the antigen-presenting cell that initially takes the food and presents it to the big orange ball in the center, which is the T cell. And that T cell then interacts with the B cell, which is the blue ball, that produces the antibody that causes allergy. And the environment with which these 3 cells interact determines whether or not you get a protective allergic response or you have an allergic response that leads to allergy. So what we really need to do is change the environment that these immune cells work in to switch them over from an allergic type response to a more protective life response, which is the natural sort of way things have done for eons before recently. Now, the next slide shows that many different types of T cells can be generated by this environment. And the naive T cell, the blue cell in the middle, can go either towards allergic reactions or can go, which is in the lower right panel, or can go towards inflammatory reactions, which is T17 in the upper left panel. It can go into a regulatory state, which may be helpful in inducing tolerance, which are the Tregs in the lower left panel. Or you can go into a cellular response, which is needed to protect against viral infections. So the real idea here is by changing the cytokines that are produced, the hormones that are produced around these T cells, you can drive them into different types of allergic response. So you can see here that the allergic or Th2 cell is generated by 3 cytokines, IL-4, IL-2, IL-13, and IL-2. And if we could block those, we could prevent the development of allergy specifically while still letting other types of immunity persist. On slide 31, this is an article from Sally Wenzel at Pittsburgh. You could see that the pharmaceutical industry is hard at work trying to develop things to block allergic reactions specifically. And of course, this slide is related to asthma because most of the drugs are being developed for asthma because it's an easy endpoint. But all of these markers actually pertain to food allergy as well. So we've learned a lot from trying to treat asthma about ways that we might go about trying to treat food allergy. And on slide 32, I just list a number of targets that have been developed by the pharmaceutical industry that may be useful targets to prevent allergy to food. And you can go through this yourselves. The reference to Sally Wenzel's articles on here, but suffice it to say, we do not lack for drug targets in food allergy. What we lack is drugs that address those targets and are being tested for food allergy. So there is good news. And the first bit of good news that's coming out right now is a new drug called Dupilumab. It's actually an antibody therapeutic from Sanofi Regeneron. It's in final testing for atopic dermatitis. And this is really a wonder drug in the clinical trials for atopic dermatitis. Young people who'd never had clear skin within 30 days were cleared. And in fact, 90% of the people put on this were cleared as compared to 15, 20% on placebo. So it is pretty amazing for atopic dermatitis and should be approved within a year for this application. Remarkably, when it was given to people with atopic dermatitis, it also cleared their asthma and their allergic rhinitis or their nasal allergy symptoms. So this is something that we believe could block out the entire allergic response. And so we're very hopeful that once this is approved for atopic dermatitis, with many food allergy people have, it may also play a role in treating food allergy, and we look forward to that. The next slide shows you how this works. It actually binds and blocks to the receptors for those hormones that mediate the allergic reaction, IL-4 and IL-13. And so it's actually remarkable because it shuts off the specific signals for allergy without harming normal immunity. And this is obviously very important. You don't want to make yourself susceptible to infections the way you do with steroids or other non-specific drugs. This only affects allergy, but it affects all types of allergy. So we're very excited about this drug and know that it'll be out within a year. So finally, one of the things I'd like to talk about is how do we improve the quality of the life and health of individuals with food allergy. We know that we're working on therapies to try and help food allergy. But in the meantime, we'd also like young people to really live a quality life, to be safe and effective. And one of the problems we have with food allergy is that there's no definitive governmental statement about its importance. And we viewed that as a big problem at FAIR. And so we funded an Institute of Medicine consensus study. Now, the Institute of Medicine is the government's arm of medical research. It was founded by Abraham Lincoln and basically sets the standard for medical problems within the United States. We actually instituted and organized and sponsored a study on food allergies, their global burden, causes, treatment, prevention, and public policy. So this will define everything in terms of the importance and public impact of food allergy, and should be able to put a dollar amount on how food allergy alters and really costs people who have it. The study will seek to document the burden of food allergy in the American life and its role as a public health issue. And there'll be a comprehensive report this coming year from the organization. And these findings will provide essential information that should help to guide further research, education, and public policy efforts, and really change how we think about food allergy, not just within the community of people with food allergy, but among the non-food allergic population. And this is very important because we need to have them make accommodation for our food allergic individuals. So finally, I revert to our mission statement at FAIR: life, health, and hope. We hope to support the life of food allergic people and allow them to live safe, productive lives, and to get them the respect of others so that they don't have to be afraid, that they aren't bullied, that they really have a high quality of life. We also want to enhance the access to state-of-the-art healthcare for people with food allergy. And our clinical network should help with that. And finally, we want to provide people hope that the research that's going on now will provide new therapies, and therapies that will be broadly applicable, not just to one food, but hopefully to many. But we have a long way to go. And as a reminder of that, I do one thing every month that I find very difficult. I write personal notes to the families of young people that have died from food allergy. And sadly, I had to do that every month this year. And in fact, slide 39, where I'll end, is the listing of the young people that died in the past 13 months or so from food allergy. You'll see that many of them are actually in their adolescence, and they're out enjoying life for the first time, and unfortunately that can put them at risk. We need to make these young people safe. We need to find the therapies that will keep them from having these reactions. This is a tremendous loss, and one that both I and FAIR are committed to seeing end. So with that point, I'll thank you for listening to this. And I put up just some of the website links that we have to different places on our website where you can get additional information.
All right. Well, thank you once again, Dr. Baker, for sharing all of your insights, your expertise. We did get some questions that came through, so we'll try to address as many as we can. In case you missed any portion of the presentation, though, today's webinar will be recorded and available as a resource on FAIR's website in the coming days. So just to be mindful of that, because we did get some questions for some people that were latecomers. A few questions that came through were sort of related to some of the studies that came through. Some are related to our clinical network. So one question that came through, I think partly as the results that we have seen sort of with the LEAP study, but also with Viaskin, does desensitization seem to work less well on adults or as children get older based on what we're finding, or is there not evidence to indicate one way or another?
Well, there seems to be a bit of a dichotomy. The patch works best in young people, and in fact, it only showed an effect in a group that was up to age 11. And that's why that's the group that's being tested in the phase 3 trial. That may relate to specifics of how the antigen gets across the skin or how the immune cells function in the skin of young people. OIT seems to work equally well in children and adults. There are children that can't tolerate it, there are adults that can't tolerate it, but there's really not a differential there. So it may depend exactly on what the approach is to desensitization. With OIT, it may work better in adults than the patch, but in fact, there appear to be ways to try and address this in both populations.
Great, thank you. Another question that came through was related to one of the studies that Dr. Nagler has done and reported on at our national conference this year, but they were wondering if you could provide any insights on the latest update in terms of her study with probiotics and Clostridia and where that study is at right now.
Yes, I saw Kathy down at the food allergy meetings in Miami about a week ago. I think right now all of her data is in mice. And I think it's interesting, and I think it suggests that there may be manipulations of the gut bacteria that were effective in seeming to alter the allergic response in mice. Obviously, mice aren't people, and these studies will now need to be validated in human trials to see if the same thing happens. Humans have incredibly complex, gut bacteria, and I think it'll be very challenging to do this. But certainly, before any of us can make any statement about the impact of this in humans, we'll need actual studies in humans as compared to mice.
Great, thank you. Another of our listeners asked what the best way is to search for trusted clinical trials, and will the FAIR clinical network site, will we list participant requests for our clinical network sites from FAIR?
Yes, we'll list the contact information on our website from the clinical network sites. The other thing is there's a site that at the federal government at the FDA called clinicaltrials.gov, and that lists all the available sites. So if you list by keywords, food allergy or peanut allergy or something like that, you'll get a listing of the clinical trials, and under those listings, it will define which sites are available. So yes, FAIR will help with this, but there's also a site at the FDA that you could go to that will give you information as well.
Great. Another inquiry asked, is Dupilumab being tested for children or just adults?
Right now, Dupilumab is being tested for both children and adults with atopic dermatitis. So, you know, it will be approved in both populations. It may be a while before we get down to very young children, you know, in their first few years. We'll need safety data before that happens. But yes, it's being tested in both children and adults right now for atopic dermatitis.
Great. Another question going back to some of our potential causes of food allergies. You had mentioned the hygiene hypothesis earlier, but just any general insights on the sudden rapid increase in food allergies and asthma that you might have?
Well, it's interesting right now. First off, I don't like using the term hygiene hypothesis because I think it oversimplifies this whole thing. You know, we're looking at environmental factors, you know, we're looking at food factors, we're looking at genetic factors that all come together to shape the allergic response. And clearly, there are many more people allergic. But in fact, when you look at the population of food allergic people, there are a lot of variations. There are people that are allergic to a single food. There are people that are widely allergic. There are people that also have asthma or eczema. There are people that don't. So it's probably not one thing and not one particular trigger that's doing this. I think the most important thing is that early life is probably very important. And the fact that the LEAP study suggested that if you start many of these foods as soon as you're weaning your child within 6 months or beyond that, yeah, there's a likelihood, a better likelihood that they'll be tolerant than if you wait. So I think that's probably the most important take-home point right now, at least for peanut. I think that there are lots of other things we need to look at. Clearly, we don't want to go back to the point where children were dying from infections or having severe disabilities from viral infections. They're easily prevented by vaccines. So it's not like we're going to do away with those things. Those are very valuable things that have lengthened human life. But the immune system's different now, and we need to know how to manipulate it so that we get the good effects of these things without some of the side effects.
Great. A couple of other questions that came in through about Dupilumab. One was asked if there is any indication in the study that the drug might have any effect on eosinophilic disease, such as eosinophilic esophagitis for food allergic individuals, as well as are there any negative side effects that you're aware of?
Well, I'll answer the second question first. It was actually remarkable. There were fewer side effects in the treatment group than the control group. And that was because the number of skin infections dropped dramatically once the skin had cleared from the eczema. So in fact, this is one of the first studies I've ever seen where the control group had a much worse side effect ratio than the treatment group. So that's all good news, and it argues that this might be a very safe drug. The second thing, which I think is very important, because this seems to fundamentally alter the allergic response, there is hope that it'll be effective for a multitude of allergic diseases. And in fact, the idea that you could treat the skin and the asthma gets better might also lead one potentially to hope that eosinophilic esophagitis gets better. And we're very hopeful that for this very difficult to treat disease, Dupilumab gets evaluated fairly quickly for it, because if it does work, it would provide the first real therapy for eosinophilic esophagitis. And if it worked as well as it does for atopic derm, it could be a remarkable treatment for that disease.
Okay. At this time, we are coming up to the end of our hour, so I wanted to promote our next webinar. If you have questions that were not addressed here, please feel free to email us at education@foodallergy.org in case there are any questions that have not been addressed here in the audience setting. But with that, I want to just promote our next upcoming webinar. And also to let you know that after this webinar, we will be following up with an email that will include a short survey link, and that will be included for you to share feedback on today's presentation, as well as to provide comments on webinar topics that you would like to see in the future. So if you could, please just take a minute to complete this and help us continue to deliver meaningful information to you from all of us here at FAIR. All of FAIR's resources, education programs, and a number of the research efforts discussed today are really made possible through the support of our community. And I wanted to let everybody know that through December 31st, all donations to FAIR will be doubled thanks to a matching gift of up to $100,000 from FAIR's Board of Directors. So if you would like to make an end-of-year tax-deductible gift to enable FAIR's mission efforts, you can donate online at foodallergy.org/donate. Next month, we will be joined by Gwen Smith, who is the editor and co-owner at Allergic Living magazine, and she'll be discussing onset food allergy, adult onset food allergy. She will be sharing insights from her personal experience with adult onset food allergy diagnosis, including some lifestyle impact information and how to adapt, as well as some of the current findings on adult food allergy from a research perspective. This webinar is part of FAIR's Adults with Food Allergies webinar series and is made possible with generous support from Mylan Specialty. So at this time, that will conclude our webinar for today. I'd like to thank all of you for joining us, and on behalf of all of us at FAIR, I'd like to wish you a happy holiday season and a new year. We hope to see you again in January.